Thank you. Thank you. That was, I think, an introduction that just, I think, was exaggerating things. But we certainly, like IU, are making an effort to, do our bit for myeloma. And I think all the larger centers, academic centers, I think, are worthy of praise here. So I have been given the, talk to discuss the role of CAR T. And obviously, CAR T is something that I presume most of you are aware of, is a modality of therapy that really is going to be changing the landscape of treatment for not only myeloma, but a number of hematological cancers, and I think possibly even some solid tumors. So we're going to just have a little overview first about the CAR Ts. So CAR Ts are natural immune cells that are modified to attack the cancer. So this is just giving you, okay, sorry, what did I do? I kind of probably reduce the size. Okay, there goes. Sorry. So as far as CAR Ts go, it is, as the name suggests, CAR stands for chimeric antigen receptor. So what is usually produced in a laboratory is then transduced into the immune cell. So I think the next slide will give you a better idea of what this involves, but the CAR construct has an outside the cell portion called the antigen binding domain. It has then got what we call an intracellular domain, and these have distinct functions, and these are borrowed from antibodies, their basic structure. Oops. So what goes on when we are talking about doing a CAR T is first we collect the T cells from the patient. This is done by a process called pheresis, which is something that some of you may have undergone for even stem cell collection. It's the same machine, however, it's a much simpler process. You don't have to get growth factors for 5 days. It is just that you get date assigned and you show up. And instead of usually in most centers, what's 4 to 5 hours on the machine, it's just 3 to 4 hours on the machine. And this machine is set such that instead of collecting the instead of collecting the stem cells, you're now collecting immune cells. These immune cells are then shipped to usually a pharmaceutical company. At the moment in myeloma, there are 2 of them, Bristol Myers Squibb and Janssen. These are shipped to their facilities where these cells are then modified. They're using what we call viral vectors. That CAR construct that I showed you on the first slide is then introduced into the immune cell. The cells are then expanded. They go through quality assurance. And usually it's about 4 weeks before they've gone through all the manufacturing steps. At that time, the center is informed about the cells being ready, and then you generally have to give 3 days of chemotherapy, which is not as intense as chemotherapy given for purposes of stem cell transplantation. It's moderate intensity therapy, doesn't make people usually very sick. And usually 2 to 3 days later, you get the CAR cells. In most centers, the CAR infusion is being done as an inpatient, but I'm told here at IU, you're already far ahead of us, where we are planning to do it as an outpatient, but haven't yet worked out all the kinks to do that. So if it is done as an outpatient, it's usually that the patient can stay in the vicinity and sleep if they're in town in their own bed, or if they're coming from far away, they have to still be within 30-minute drive of the center if it is being done as an outpatient. And the reason why patients are in the hospital or in the vicinity is because these cells, once they recognize their target and they start multiplying, they secrete chemicals that make people usually run a fever. And then what also happens often is a drop in the blood pressure. which is usually mild, or a small need for some oxygen. It is rare for patients to require ICU level care. The other thing is a period of confusion can also ensue. This is something that is also usually self-limited. In a case of CAR T cells in lymphoma, some of these can be much more profound than what we have seen in the case of multiple myeloma. The cells are then, going to do their job over the next several weeks. Patients are in the hospital, in our hospital, where patients are hospitalized for usually anywhere from 10 days to 2 weeks, and then they're discharged. After discharge, patients have to be on some immune suppressive, on some drugs to ward off infection due to the immune suppression that the patients unfortunately develop with the CAR product. And also several patients do require to get immunoglobulins on a monthly basis because these cells are so effective that they eliminate the cancerous plasma cells, the myeloma cells, but they also eliminate the normal plasma cells. They can't distinguish between the two. And over time, the normal plasma cells return and the immune globulin levels can go up. So right now, CAR T cells are pretty much in later lines of therapy. Usually you have to be through 4 different lines of therapy and progress through 4 different treatments before you can get a CAR T. And because of the cost of these, the insurance companies are putting up more and more barriers to be able to justify that a patient is a candidate for a CAR T. In the past, when we used to give drugs for patients, they used to just believe us and say, okay, just tell us that the patients had 4 lines of therapy, that's fine. Now increasingly several insurances are wanting proof that each patient progressed within each line of therapy. They want actual values, which they sit on for 2 to 4 weeks often before giving approval. The other thing we've also noticed is in a few cases that they say, yes, the patient meets the criteria the FDA set up for the trial for this, but they didn't. We went back and the patient wouldn't have met the trial criteria that led to the approval, so we will not allow it. So these are things that we are facing for the first time with these expensive therapies. But I think these are barriers that will be overcome. But what we need to do is, in terms of strategizing any treatment that works very well in later lines, we know that in a much later line of therapy, depending on the CAR T used, anywhere from a year to even 3+ years, depending on depth of response, can be expected on an average with these therapies. So moving them earlier should lead to better outcomes, and that's where they're headed. So in the very near future, I think within the next year or so, we will have FDA approval to give these in patients who have just progressed once. And we have now data from 2 large trials that have shown that these are superior to our current approaches. And so it is a matter of time before they move there. And I think that they will then become applicable to even more patients, because if you wait to do anything to a later line of therapy, unfortunately, there are patients that don't make it to that line of therapy. What we also are looking at is for patients who relapse early. We know those patients who relapse within, say, 18 months of a transplant instead of the average 4 to 5 year period, unfortunately don't do well with subsequent therapies either. So giving this CAR T at time of first progression also is being looked at in clinical trials. And some of these trials have shown impressive results already. The next thing is, can we not even wait for patients to relapse? Can we try to move it into the very initial phase of a patient's treatment? And as you know, that for most people who are transplant eligible, still transplant is thought to be the standard of care. So people are looking at all different strategies to get it into frontline therapy. One is to give it to patients after transplant when they've not had a complete remission. And some of the data, again, from clinical trials is very encouraging, with over 90% of patients who got it in tandem with the transplant not having had progression at a year, which is pretty remarkable. The other thing is, can we just remove transplant from the paradigm? Could we just not even have to do transplant? And that also is being looked at in trials that have just started, trying to see if you could do CAR T cells as part of your initial therapy and not have to take patients to a stem cell transplant. So whether it is in conjunction with a stem cell transplant in first-line therapy, or it is a substitute for stem cell transplant in first-line therapy, we'll have to wait several years for these kind of answers, probably more than 5 years from today. So we do know that unfortunately the cells do stop working at some point, and there is no long-term data whether some patients never progress, but I think the prevailing thought is that unfortunately at some point most patients will break through. And why do patients break through is also the subject of great research. Is it that the cells no longer have the target? So in the case of CAR T cells that we have today, they are to B cell maturation antigen, BCMA. So have these cells lost their target? It appears that only in few cases do the cells lose their target. So that it may not be the major mechanism. Or is it that these T cells just get exhausted over a period of time? They're just not working anymore. So different strategies are being employed to actually improve T cell function and to prevent exhaustion. The third thing is a constant level of CAR T cell in the blood required for them to maintain their effectiveness? Most of the trials show that after a period of time, you cannot detect these CAR T cells anymore, suggesting that they do their job at the beginning and then they disappear, and the disease can still remain in remission for long periods of time, even if the CAR T cells are not around. But again, perhaps using very sophisticated techniques in the future, we may be able to detect very low levels of these CAR T cells, and perhaps if they are not present at those low levels, patients will relapse. We don't know the answers to this, whether improving persistence is the way to make them work longer. The field is tackling all these issues in a variety of ways. As I said, one thing is to look at new targets beyond BCMA, and we know that CAR T cells to these different antigens, CS1, CD38, and GPRC5D are already in clinical trials, and some of the results from the GPRC5D clinical trials are pretty impressive. Even patients who have progressed after BCMA-based CAR T cells are responding to CAR T cells to GPRC5D. Other ways are to combine two CARs. Perhaps in the future you could have a BCMA CAR and a GPRC5D CAR, so that you can have better results long term, that even if the cell loses one receptor, you could have the other still present. So that, and then people are actually trying to put two CARs into the same cell. So instead of having one CAR to GPRC5D and one to BCMA, just have one cell which carries the CAR receptor for both. So a lot of different things are being done. People are trying to combine these with drugs that several of you may have been exposed to, like lenalidomide. They boost the immune system and potentially could boost the CAR T cell activity. So one way is to see if you can give lenalidomide after the CAR cells to improve their effectiveness and their longevity. So all different kinds of things are being tried. That is just one example out of many. The other way, obviously, is to see if we can make the process simpler by doing what we call off-the-shelf CAR Ts, or what in medical lingo is called allo CAR Ts. We know that the process of generating the autologous CAR T cells, as we said, is a little cumbersome, and it does take a period of time. It takes up to a month, as we said, for after the pheresis for the product to be generated on an average. And before that, you have usually a month or several weeks fighting the insurance to get it. So you do often have to wait 2 months or more from the time you first see a patient before you can infuse the CAR T cell. So having an off-the-shelf product which is generated, what is called an allo CAR T, that is another way forward. People, I won't go through the minutiae on the table, but just to say that these are already being looked at in clinical trials and have proven effective. What we don't know is how they compare in their effectiveness and their longevity compared to the autologous CAR T. But if they succeed, that will make the process very much simpler, in that you won't have to be generating a CAR T for each patient and waiting a month. You'll just be able to reach off the shelf and get it. There are obviously some technical challenges around this with graft versus host disease and rejection that we have to fight, but I think we probably will be able to overcome that. The other thing is, as we just discussed, moving it to the outpatient setting will be making it easier for the companies and for the hospitals. The economics of CAR T cell also favor outpatient administration. I won't go into the details there, but we're glad to address those questions subsequently as to why outpatient CAR T cell therapy is economically more feasible. And then there are a whole bunch of other kind of immune cells, not only T cells, there are other cells that are also immune cells in the body, the NK cells, the NKT cells. All these have also been looked at now in early phase studies to be able to serve as the cell subtype in which the CAR can be carried. So we'll have to see how those pan out. I think that was my last slide, and I think the questions are later.